Hello, everyone, and welcome back to our GETA Family and Professional Summit 2021. And last, but definitely not least today, we have a fabulous panel we're going to talk to and get all of their insights. Now, Melbourne, as I mentioned earlier, might have a little bit of difficulty when it comes to COVID lockdowns, but we do not have a difficulty when it comes to recruiting the best in the world when it comes to epilepsy. So I'm going to introduce you to some of our homegrown stars here today. I'm going to start with Dr. Katherine Howell, a pediatric neurologist and epileptologist at the Royal Children's Hospital in Melbourne. She's also a clinician scientist fellow at the Murdoch Children's Research Institute, among many other things. Now, Katherine's particular area of expertise that we might want to hear about from her, among others, is SCN2A, and I'll ask her more about that later. Then also here today, and this is on my screen, yours may be different, I'm going to turn now to Professor Ingrid Scheffer. Professor Ingrid Scheffer happens to be our daughter Jackie's doctor, but that is just one reason that I know her. She is a trailblazing scientist and leader who is known around the world for her breakthrough concepts about genetic epilepsy, including the very first one with her scientific partner, Sam Berkovic, that non-inherited epilepsy could indeed be genetic, which started the ball rolling, ladies and gentlemen. It's won her a lot of prizes: Order of Australia, L'Oreal, UNESCO Women in Science Award, Prime Minister's Prize for Science. But what we're going to talk to Ingrid about today are some of her newest research things today, which, for example, a clinical trial with Xenon, a natural history study. More on that coming up from Ingrid. Now you, Steve, going to the hot seat. Steve is the director of the Florey Institute for Neuroscience and Mental Health. Also in, by the way, Melbourne. Are we noticing a theme? Steve also has a lot to say about SCN2A. And that is by no means all. He's acutely aware of how to turn discoveries into therapies and treatments. And I know that is so important to everyone here. Steve is the Chief Scientific Officer of Rajkan Bio, which is pioneering breakthroughs for epileptic encephalopathies caused by SCN2A mutations. And I've mentioned SCN2A a couple of times now, and you're about to hear it a third time. And I want all of you who do not have a child with this mutation to hang with me. And what I like to think of is we're a bit like the Three Musketeers, you know, all for one and one for all. A breakthrough for one of our number is genuinely a breakthrough for all of us. And so it's thrilling that we're seeing some good breakthroughs on SCN2A. They aren't the only one, but we'll be talking about them. That takes me to Chris Pierce. Chris Pierce is a rare disease advocate, a health professional. She's one of the co-founders of GETA, and she has a son, Will, along with David Cunnington, another co-founder of GETA, and their son, Will, has SCN2A, so she knows a lot about that. She's also on the board of Rare Global Advocacy Leadership Council at Global Genes, and we mentioned this earlier too. Chris recently started working at the Epilepsy Foundation, which is a huge win for all of us in the genetic epilepsy space, because she's going to be making sure that we all know what's happening there and help us with some of the latest breakthroughs that are coming too. And last, but most definitely not least, I bring to you Danielle Williams. I happen to know that her husband, Danny Williams, is very nearby. Who knows, he might chime in during this call. Danielle, another co-founder of GETA with her husband, and she's the founder of SynGap Research Fund in Australia, a co-founder of SynGap Global Network, a co-founder of GETA, and she and her husband, Danny, are the parents of Jaylee and Dolly, who have SynGap epilepsy. One more thing to know about Danielle, and I want to talk to you about this, Danielle. She has lobbied governments and raised $2 million for genetic epilepsy. That is no small feat. So, and as you know, I have a daughter with KCNQ2. So there's a lot of different genetic mutations just within us, those of us who started GETA, and most importantly, a wealth of knowledge from our scientific panel. So I said we were going to talk a lot about SCN2A. If I may first go to you, Steve, and ask you about some of the latest developments. You've got some treatments that are just going in right now and some work with Praxis Medicine, among others. Tell us what's happening with SCN2A and why everyone who's joining this call should be excited about that.
Well, SCN2A development of an antisense oligo for patients with gain-of-function mutations is well underway. There are activities happening now. I mean, some of the things you have to do before you put it into people, you have to understand whether the safety profile. You have to understand how much drug you need to give to a patient. You have to understand what you're going to measure, and you touched on that earlier, Sara. So some of those very, very important activities are being undertaken now, so that we can be best prepared when the first trials do happen, that we are can conduct them in the most efficient and hopefully successful way. Unlike many other drugs which first go into healthy volunteers, drugs such as antisense oligos that are for rare diseases have to go directly into patients. So understanding which patients need to be targeted for these drugs is a really, really critical aspect of what we're trying to do. And we're hoping in the not too distant future that we'll start to make announcements around trials in this space.
And do you have any, I want to push you on that, do you have any idea when those trials might occur? Are we talking this year, next year, 5 years, next month?
It's well within 5 years. It'll be, within a year or two is what I'd like to leave it in, that sort of time frame. You never know what's going to happen along the journey. That's all, and we don't want to give too much false hope.
Yes, I understand that, Steve. Let me ask you just before we move on, and I want to stick with this SCN2A theme for a moment, but ASOs, we hear about them all the time. Some people on this Zoom cast are very familiar with them, and some it's going to be new to them. Can you just give us a very quick précis on what that is?
It's very similar to the technology, or identical to what Steve Wilton was talking about for the muscular disorders that he described. It's a medicine that can directly act on the RNA. There's a thing called the central dogma. You have your DNA, which is your blueprint. You have your RNA, which is your working plans, and you have your proteins, which is the house. This medicine works on that intermediate working plans, and we can either shut them off, we can modify them, or we can turn them on. And that's the ASO medicines. Let us do that with quite a bit of control, which is why we like them, because the control is critical for the sorts of disorders we're working with, where we want to specifically do a thing to the gene.
All right, let me move, if I can, now to you, Katherine Howell. We're going to talk a fair bit about this and unpack it if we can. What is your role in this project as we're working on not just SCN2A, but other DEEs that might be affected?
Yeah, so quite a lot of what I'm doing in the SCN2A space, which is actually really important for all genetic epilepsies, is looking at the natural history of the condition. So what are the specific clinical features and how do they evolve over time? And that change over time is really important to understand what we expect to see in a person at that particular stage of their life. And that's so important, not just to be able to tell people at a particular stage in their child's journey what to expect for the future, but for when we are looking at trialing new medicines, to understand whether they are actually altering the natural history of the condition. And so, for example, if a child starts a new treatment and the seizures stop, have they stopped because of the medicine or because that's the age at which they would have stopped in that condition anyway? And Joseph touched on that before. With the natural history studies, as well as understanding the spectrums of the condition, we are also looking at what would be the best things to measure as trial outcome measures. And you touched on that a little bit before. And that sort of goes to the questions of what are the most problematic aspects of the condition. Very often it's more than just the epilepsy. In fact, in our SCN2A study, we asked families which were the most problematic symptoms, and only 8% said seizures were the sole most problematic symptom.
Just to stop you right there, Katherine, I think that's one thing that continually just kind of hits me when we have our KCNQ2 meetings, and that is that when we have new scientists are coming in, they're talking all about the epilepsies, and when we hear from families, although some percentage of the population cares a lot about that, of course, but others, that is just actually walloped by some of the other things that they're dealing with. So is that pretty common among a broad range of the DEEs that we're talking about here?
100%. It's extremely common. In fact, I'd go so far as to say it's just about universal. And we also think about sort of development and cognitive function being a major thing, but actually really big things for families in SCN2A and other conditions include things like behavior, sleep, irritability, things that might impact your mobility, like dystonia or other tone abnormalities. And so, so we're trying to work out what are the problems, how could we measure them? Are there existing tools to do that? Do we need new ones? And those kind of things will be guided by the information we get from the natural history studies.
And when it comes specifically to SCN2A, how, all of the people in this community, they have their individual area that they're concerned about. How significant do you think the progress is with SCN2A when it comes to some of the other genetic epilepsies that maybe aren't so far down the track? Is what's happening with SCN2A a harbinger of things to come for some of the others?
Look, I think so. I think that a lot of the lessons we're learning from SCN2A can be applied to other conditions. I think Steve's work in SCN2A and the laboratory clinical collaborations that have been done over time to help correlate some of the findings that Steve's made are really critical. I think that's been something that's been done quite well in SCN2A and is actually also sort of now being done in a lot of other conditions. But that someone mentioned earlier, things like gain versus loss of function. So a lot of the genetic disorders are not just one condition. They're actually two or more distinct conditions due to the same gene. And you're going to need different treatments for that. The treatment for one might make others worse. And trying to tease those aspects apart is really important too. And that's something that Steve has done very well.
I think that's important to make that point. I'm going to come to our advocates in a moment, but Ingrid, I want to come to you as the third scientist here on our panel. Katherine was just talking about natural history studies and the importance of that. I know that's of great importance to you. Can you tell me what you're doing when it comes to natural history studies?
Well, the good thing is I'm working with Katherine and with Emma Palmer in Sydney and hopefully the Australian Child Neurology Network, but very happy to take it more broadly to people around the world. And that is really to make a gene agnostic, which means we don't care which gene you have and we don't even care if you don't know your gene, but we're interested in your natural history of the child or the adult with the disorder. And just to expand a little on what Katherine was saying, the comorbidities, which means is medical term for other features, which can be movement disorder, it can be how they walk, it can be how they talk, it can be their gut problems, their chest problems, their spine problems like a scoliosis, it can be any number of things, heart problems. They often end up being a major problem to families, but different diseases have different problems. And just to illustrate the big picture here, we have more than 300 genes now for the developmental and epileptic encephalopathies, which is 300 different diseases, if you just think of it as a gene. And then within the gene, they all have a spectrum, as Katherine said. In the simple sort of understanding of an ion channel, it might be gain or loss of function, but then there can be a whole spectrum along there. And if you use a treatment to stop a gain of function, as Steve is developing in SCN2A, you want to get back to the middle land. You don't want to get down to this end because that also causes autism and intellectual disability with loss of function. So the natural history is about mapping out the natural history of that gene, if you know the gene, and if you don't, we'll have the information ready for when you do, and then figuring out when seizures stop, so that, for example, KCNQ2, the seizures might stop when you're 2, but you may develop other serious problems. Whereas other genes, the seizures may never stop. SCN1A, Dravet is a good example of that. So you have to understand that natural history, so that if you come along with a Steve Petru ASO, you can actually say, does it really change the outcome for that child?
You know, Ingrid, let me ask you, because you were there at the beginning with this, with that whole concept that non-inherited epilepsy could be genetic. Did you have any idea that fast forward and you'd be looking at 300 plus genetic epilepsies?
No, I don't think we had any idea. And when I trained, they were all symptomatic, which you had anybody with intellectual disability and epilepsy was really thought to be an acquired disease. So it's completely reframed it. And I'm really excited by the young pediatric epileptologists, epilepsy specialists who are focusing on genetics like Katherine, and also geneticists like Emma. They don't grow on trees, those sorts of geneticists that really care about the whole picture. And I'm really excited to have these people that are going to develop this a gene agnostic natural history study with us, because it's a big picture, hopefully long-term endeavour.
Well, I think that's very important. Before I leave you and go to our two advocates, I want to ask you one more question, sort of hot off the presses. I know a lot of people on this Zoom cast are very interested in clinical trials and what might be upcoming. Do you have any in the works? And if so, can you tell us about them?
Yeah, look, we've got about 15 trials going at the moment, and mostly for very boutique diseases. So that means like we're looking for 2 or 4 patients with trials in those diseases. If we get more, great. But each of those trials is a huge amount of work. We have a big trials group of 6 nurses, and some are particularly pediatric. So the one I'll mention, which I know is close to your heart, Sara, is KCNQ2, which is a potassium channel gene. And there's a study called EPIC, E-P-I-K, because it's a potassium channel is a K. And that's a randomized controlled trial for 1 month to 6-year-olds with proven KCNQ2 developmental and epileptic encephalopathy. And that means we know the gene abnormality and we know that it is associated with loss of function. And this is sort of a precision medicine, but not a gene therapy. So it's a particular drug which opens this potassium channel, which is not working properly, and tries to make it work better. The downside of a trial is that you might go into the placebo arm, and that means you're getting effectively sugar tablets or something with nothing in it, and that may do nothing for 3 months. And you say, well, why would I bother doing that? And you bother doing that because at the end of that 3 months, your child actually gets the opportunity to trial a drug. And maybe that drug will be transformational. Maybe that drug will come with bad things too, and nobody can promise you that it won't, except that it's already been through several stages. So I think that's very exciting. It's happening in several of these, or many of these, rare diseases now. And I'm really pleased that the companies, which are often based in the US or Japan or other places, are coming to us because we have so many people who are experts in this field. Can I talk about another?
And if people want, yeah, you can.
Yeah.
I was just going to say, before you go to the other one, if people want to sign on for any of these clinical trials, can they reach out to you? How do they do it? And I'm not just talking about this one. If they have interest in clinical trials, how do they reach out?
That would be wonderful if you contact us at the Epilepsy Research Centre at the Austin, and send an email. I think it's epilepsy-austin at maybe austin.org.au, I don't know. One of my team, if you can put it in the chat, I'd be very grateful.
I'm sure we can put that in the chat. We'll make sure we get that.
I ask the companies always, will they fly patients down? I've just seen Jenny has asked that question. The problem with the trial is you have to be having 4 seizures a month for that trial, Jenny. So if you're not having 4 countable seizures a month, they won't include you, which is frustrating, but that's the way these trials work, and that's because they're all dollars based. And if you have a shorter trial period, you're more likely to show it's more doable.
All right. And you said you had one more thing you wanted to talk about before I move on.
I just wanted to talk to the Dravet world because Steve is really leading the SCN2A world with Katherine, and I wanted to talk about Dravet, which is, as many people will know, SCN1A, so it's a different alpha subunit. And there are actually 2 gene therapy trials going ahead in that space. One is not coming to Australia at the moment, and I've forgotten the name for it, but the company is Stoke Therapeutics, and they are using an ASO a bit like Steve described, but this time instead of bringing down the channel function, it's trying to, you've got a bad copy and a good copy, it's trying to make the good copy overperform, if you will, and give you a whole lot more SCN1A. So they have started in the US, and so the data is being obtained. Those sorts of ASOs, I understand, will need to be given every, say, 4 months, a bit like SMA that we heard about earlier from Steve. The other sort of gene therapy is one being developed by a company called Encoded. They have a study called Envision, and they are looking at a gene therapy that again goes to the good copy of SCN1A, and it's basically turns up the starting function. It says make more of this. And my understanding is that's with an AAV, which is a viral vector, so a virus takes it into the cell, is that this would be a one-off treatment. Much more expensive. They're all expensive and much more expensive, but we don't know if it will work yet. So just to say there are many different ways we can approach these diseases with gene therapy.
All right. Well, that's fascinating. And as I said to everybody, there's a lot of hot off the presses information in this particular chat, and you've just heard some of it there. Now, if I can slide over to you, Chris Pierce. Chris, as I mentioned, is one of the co-founders of GETA, my colleague here. I just wish we were all actually physically together. But Chris, you have an exciting new job with the Epilepsy Foundation. Can you tell us a little bit about what you're going to be doing with the Epilepsy Foundation and how that really does affect everybody who's on this call, at least everyone in Australia and potentially beyond as well?
Yeah, thanks, Sarah. So I guess I just want to acknowledge that this role has really come out of the work of all the people sitting in this room, that we've been able to highlight that the families impacted by DEEs need more support. They need more vision amongst the community, and we've been able to build that acknowledgement over the last 5 years that GETA has been around. So it's basically, we've been able to work with the Epilepsy Foundation, who have always supported GETA, to acknowledge that we need to put some resources into this area. So it's a great start. The things that I will be focusing on is basically, like to Ingrid's point, it'll be gene agnostic. My job will be to make sure that all genes have a voice through the work that I'm doing at the Epilepsy Foundation. We'll be doing that through developing resources. We'll be signposting to research, whether it's what Emma Palmer's doing in Sydney and her team with cogenes, whether it's the work that Katherine's doing, whether it's what Steve's doing. Like, we'll be basically a hub for all that kind of information, and pushing people out to connect and create opportunities for collaboration. We'll be running education programs around what a DE is and why that looks different to epilepsy without those comorbidities. So we'll be looking at educating healthcare workers, families, schools, and we'll be developing modules around that for families to be able to share with their communities and family members. We're going to be setting up a national consumer advisory group so we can bring in people from different genes across Australia, so that we can hear the voice of a wide variety of families who are at different stages. So I hope to get that out in the next couple of weeks so we can get that going and get a lot of different voices to make sure that we are representing nationally, but also what we're doing is actually being shared nationally. The other thing, to recently I spoke at an inquiry around access to clinical trials and what needs to change in that space. So we will take on a really big advocacy role for for DEEs in Australia and make sure that politicians and people with the purse strings know that our families need support and that they need funding behind that support. So that's kind of a snapshot of what we're going to be doing, and there'll be a whole lot more things. And I guess to people in Australia who are on this call or view this later, if there's things that you see that are missing or doing well, please reach out and let us know, and we can work on it together.
Chris, just following up on that, Ingrid, is just pressing Ingrid on the upcoming trials that she's got going on, and it's quite a few. Is there a way for people to reach out to you directly through the Epilepsy Foundation if they want to get to Ingrid, if they want to get to Katherine, if they want to get to Steve? In other words, when you say you want to be the hub, is that a secondary way, or even indeed the primary way, for people to be connected to the research that could potentially affect their child's life?
I wouldn't say it would be the primary contact for research. I hope that's the answer. Ingrid and
Good. No, tell me. You'd be secondary then, in other words, you're the backup?
Yeah, I just think we would be this signposting to Ingrid's research or Katherine's research. So this is what's happening in the KCNQ2 space. This is what's happening in SCN2, and it'd be a click through to the work that they're doing. I don't think we will be controlling those strings at all. It's just really, hopefully it becomes a hub that people feel confident that that's the most up-to-date information to get to where they want to go. So for example, I'll give you an example. Over the last couple of months, I've been reached out to by industry around the Dravet trials. I've been reached out to by people around infantile spasms. Where do they go? Very rare genes which are in that 300 genes that Ingrid mentioned, and how do I connect them with? So it's just kind of an entry point that we would then connect people when and if we can.
Okay, that makes a lot of sense. Let me just quickly ask you, Steve and Ingrid, if you have any thoughts on that. If people want to be, I mean, one of the ways in which I've always seen GETA is a way to connect people to things that are happening right at the moment in which they're occurring. We've seen some connections that have happened during this call. So if anybody on this broadcast wants to connect to either of you, Ingrid, you've given a shout out. Steve, any way that they should get in touch with the Florey regarding what you're up to with SCN2A or any of the other research that you're doing?
I mean, we're always happy to hear from parents and families to share in knowledge and information to see if they can participate in any of the preclinical studies we're doing. With respect to clinical trials, the Florey, we're not directly managing or running those, and I'd direct families to Ingrid or Katherine around those. But we do like to hear from parents who are interested in participating in the earlier stages, in the ideas generation, and not only in SCN2A, but for other disorders, so that we can start to get an understanding of the disease, start to think about how we might produce the therapy against those.
All right, Ingrid, anything to add before I go to Danielle? And I'll be coming back to everyone, just so you know.
I think that families make such a huge difference. This is testimony to this, and I'm thrilled that it's been going 5 years and may go another 500 years. But just to say that Steve's initial interest in SCN2A was partly through a little girl who died with SUDEP with SCN2A when she was 21 months old, and the family came to Steve and have worked with both of us since. And I think that you just cannot believe how much impact families can make to both doctors, to scientists, and to politicians, and to their communities.
Yes, I can think of many examples of people who do this. My friend Kelly Cloesy is one, and my friend Danielle Williams. I want to come to you now for all the work that you've done. Danielle, for those of you who don't know, works for AIA Vitality Australia, and that puts her in a position where she can work on behavior change. How do we change our behaviors to change our lives? And that's an exciting concept. But in her spare time, Danielle and her husband Danny, as I said, they raised two beautiful girls, Taylor and Dolly. And your girls have SYNGAP, and you have become quite an advocate, Danielle. I wanted to start, if I could, with the money that you raised, $2 million. For those who are just starting on their journey, how do you go from, I want to help my kid, to, I just raised $2 million?
Well, Ingrid touched on it a moment ago. She said sharing the families and the children, their stories is quite a powerful motivator for politicians, for researchers, for scientists. So I think that helped. We actually had an audience with Greg Hunt, who was very kind and generous, and he tried to point us towards the schemes that we could access funding from initially. Medical Research Future Fund was the first one he suggested that we try and get some funding from, but we just weren't eligible for any of the existing funding schemes. So it ended up that we did continue to hassle him, and I think the only way that he could see the back of us was to agree to funding some research into genetic epilepsy, and from that we got some funding for research at the Florey Institute with Steve. Steve's got a lab there. So we're very grateful for that, and I couldn't advocate more for the fact that families can be quite powerful. And I feel grossly unqualified to sit on this panel, especially when there's experts in genetic epilepsy, because I'm not working day in, day out, but I am an expert in two little girls who happen to have
two beautiful girls.
Yeah. So
I do not do that to yourself, Danielle, because what I was going to say to everybody next is you wrote something that just touched me so much, and I hope everybody can find it online and read it. And it's about how you turned a tightrope into a runway, and it's a beautiful piece. I know, Danielle, I'd like you to talk about that. I know you've always been really good at being able to compartmentalize work over here and life over here, and you got to a stage where that wasn't possible. Can you share what that was like and how you decided you had to integrate the two?
Yeah, you should have forewarned me here, Sarah, because you've got me on the edge of reliving that experience.
I'm so sorry.
It's all good. Yeah, so I did, when the girls were young, firstly, we didn't know what was wrong with them. So we had a long diagnostic journey. We had maybe 5 or 6 years of trying to search for an answer. And I didn't know what to tell people initially at work. And also I didn't want people at work to think that I couldn't handle things, I couldn't cope. And I didn't want them to treat me any differently. So I just didn't say anything. And I learned the hard way that it's much easier when you're honest with people about what you're going through in your personal life, because it's not that they'll treat you any differently, as in won't expect as much from you. And I found quite the opposite. People expect the same from me, but then they're a little bit more lenient on my personal circumstances. You know, if I can't work of an evening, if I have to, then that, you know, is, if people are a lot more lenient in ways that have surprised me. So it was an interesting journey for me to kind of combine the two. And I've now, as I think you've mentioned already today, Sarah, I've leveraged my personal skills and my professional skills in this SYNGAP world as well. And what I do professionally is relationship management and strategic, combining different companies together and getting relationships built. And that's why we set up the SYNGAP Global Network. I wanted to make sure that everyone's not just running off doing their own thing in their own country. I wanted to make sure that we understood what everyone was doing. So we're not the specialists, we're not the professionals, but we can at least coordinate and say, well, this is the specialist and the professional in Australia. And those two are Steve and Ingrid. And they're doing this type of work. And the natural history study is something that's going to be global. So we want to make sure that the Americans, if they're doing a natural history study, that we can then feed that into Ingrid's natural history study, or vice versa. So my skills at my work is something that I can transfer. They're transferable skills into this world as well. And I'm hoping that it makes an impact for SYNGAP and hopefully for other genetic epilepsies as well.
Let me move then, if I can, to you, Katherine, because when, we've been talking a lot about parents and the roles of parents and really listening to all the different perspectives on the subject, that it's not just the purview of one group. There was a 2019 study that you did, a roundtable study, where you brought a lot of voices together in a room to kind of listen to all of that. Can you tell me about that study, why you did it, and where it sits now?
So actually that was something that Chris and Geta led, but we had the Geta team, Epilepsy Foundation, neurologists, geneticists, other health professionals, and a number of other parents of children with genetic epilepsies, and we got in a room together and basically sort of asked, what are we doing, at a clinical level as support of children with these conditions, support of their families, best management. What are we doing well? What are we doing terribly? What do we need to improve? What are the priorities? And got some thoughts around that. And then from that, which has taken a lot longer than I had hoped, but is very, very close, we have developed a survey, which is hopefully only about 2 or 3 weeks away before we can advertise it to people, that we would like parents of children with DEEs or genetic epilepsies to consider completing. And it's basically going to look at it's sort of, I guess, got some overlaps with some of the things Emma's doing, but is focusing more on the medical and health side of things for the child. Which particular health resources and services are you using? How easy are they to access? What's your experience with them? What are the big gaps? What are your priorities for fixing this? And also, what is the impact on the families, including some fairly simple but validated measures of things like quality of life. And the reason, one of the reasons for including those, apart from obviously wanting to understand what the impacts of these conditions are, is that in order to change practice, there are a number of things you sort of need to show, but to show the powers that be, one thing that really resonates is money. And what do these conditions cost? And it's a bit of a sort of uncomfortable thing to discuss, I guess, but it's the reality that is important. And these quality of life measures, health economists have actually sort of linked amounts of loss of quality of life, for example, to a dollar amount. And so we can start to measure how much, what are the indirect costs of these conditions on families and things like that. And we're going to use all of this data to try and inform the ways in which we can improve our clinical practice, because we know as clinicians that there are very big gaps for families. We see this with every person and family that we look after. And we want to be able to improve those. So we need to know what will be the most useful ways to do that.
Can I ask you another question, Katherine? In terms of the family piece, I know one of the things that you're doing there at the Royal Children's Hospital is you're doing some rapid genomic testing as well. Now, presumably everybody on this call has a child or somebody they love, or somebody that they're studying, if they're a researcher or a scientist, who has been diagnosed. But even today, and certainly tomorrow, there will be children who are diagnosed, or there will be children who are born who have rare genetic epilepsies, and you're working to get that diagnosis more quickly. Can you talk about that and the significance of it, please?
Yeah. So we have some existing work going, and then this is going to grow very shortly. But there is a project going on at the moment, a number of centres around the country, called the Acute Genomics project, where there is rapid genomic testing being done on children in intensive care units. That's neonatal or paediatric intensive care units. And some of our children with genetic epilepsies have been involved in that study because they're in the intensive care units, and that's predominantly the neonates and newborns with seizures, so including some children with KCNQ2 related epilepsies. And this project has allowed, you basically have the testing and you get a result in less than 5 days. Now that's really a very big shift from where things were at, where things are at in standard clinical practice now, but miles away from where they were at 5 years ago. And we are going to be expanding this study to not just children in the intensive care unit very soon, but we've now had a number of children with diagnoses made very early. This has had some very big upsides and also some real difficulties. The really big upsides are that we have an answer. We can stop looking for other things. We can stop putting a child through other invasive diagnostic tests. In quite a lot of instances in the newborn epilepsies, it actually does guide which seizure medicines we use. And we have a number of children now who have had their seizures completely controlled and are on one medicine. And historically, they probably would have been on a number and still had some seizures. And so that's really important. And I think it most likely has allowed them to get out of hospital earlier than they would have.
So that's the good news.
Yeah, obviously we have to be a little bit cautious in how much we're selling the benefits, because we're still looking at very small numbers, and this is partly why we're expanding things. But we are seeing that it informs us to do some things differently than we previously would have. The downside of it is some things that were touched on before, is I guess the sort of shock and complete change of expectations and sort of understanding of what the future might hold compared with what you were expecting for your newborn child. And that's very difficult to obviously get your head around and move forward at such a vulnerable time. But it's also, I guess, made us realise that there's also a heck of a lot of things we don't know. So, for example, KCNQ2 related epilepsies are a real spectrum in terms of their severity. Some children have epilepsy that remits and have normal development ultimately, and then others are right at the more severe end. And actually, it turns out we're not as good at picking the outcome as we thought we were. And that's also a real problem too, because giving families some certainty about what the future holds and being wrong is a real problem, or not even being wrong, but just not being able to be accurate. And so that's really driven some other work we're doing at the moment, which is sort of trying to look more clearly at early predictors of things. So it's been, in a lot of respects, it's been great. In other respects, it's shown there's some really big problems we still need to overcome.
Wow, there's so much to unpack here, and I want to come to you, Danielle and Chris, but if you will pardon me for a moment, I want to bring you back into the conversation, Steve, because one of the positives, if you will, around early diagnosis, we are told, and I wanted to get your take on this, is that in the future, as we look for potential therapies, knowing earlier might be helpful. Is that accurate? Is that a knowable thing right now, or is that another where it's going to depend? Because I appreciate your caution, Katherine, and your couching what we know and what we don't know in this emerging field. So Steve, what are your thoughts on that?
I mean, I think there's unequivocal evidence and pretty consistent opinion that for many of these disorders, the earlier you treat, the better. But the difficulty, and that's always a perspective you can have when looking back. But when looking forward from the point of identification of a mutation and then having the security of the prediction to make that bold choice is very difficult. As we know, and a work that I've recently completed with Katherine shows that even with patients with the same genetic mutation can go on and have different severity of the disorder. So, and if you knew that in advance, you may be more or less aggressive in your therapeutic choice. So the ways and the tools that we might need to develop to predict that are critical now. And because we don't, in order to be conservative and not potentially damage a child's outcome, we let a child that needed to be treated slip through the cracks, and that's not good either. So when we think about the predictive tools we need to develop, it goes back to some of the tools that Matt Might was looking at. You can't just look at the knowledge of a single gene mutation. You need to look more holistically. You need to look at other aspects of the genome. You need to look at other aspects of what we call the interactome, other things that have happened to that genome in the natural history of that particular person, from egg and sperm all the way to a beautiful little child. And so in some ways, models you can build from stem cells from patients could be useful, but they're slow and expensive right now. So as technology improves, these tools will get better and better. And then I think we'll have the courage one day to say, okay, we found a mutation. We've now checked with this other technology that this particular patient will really benefit from a therapy. Now, that in some other cases, it's going to be clear there'll be a one-to-one correspondence. Particular mutations always lead to a very severe or treatable case, and then your confidence to treat will change. All of this, with more experience, will be solved. I think we're only going to get better and better and better at doing this. And that's why you mentioned earlier, Sara, how important it is to get the first program off the ground and the second program off the ground, because the lessons that we learn from those are going to be applicable to this constellation of things we need to think about in that really important decision. The information that people like Katherine and Ingrid need to say, yes, I'm going to give this drug to this patient at this time. And that's the sort of the coalition of all these different things we want to occur.
Okay, as I said, I do want to bring Danielle and Chris back in, but just let me follow that one more second if I can. That horizon, when you're looking at the horizon, because you're right there on the kind of therapeutic treatment, precision medicine, you're right there at that edge. What are the things that you're seeing? So we're 5 years past our first GETA conference. What are you seeing 5 years from now? When we get you to present, what do you think we're going to be talking about?
I think we'll be talking about the intersection of the two technologies, of the therapy and of the diagnosis, and that's going to be that'll be the key. And that's why the work we keep hearing about, Ingrid's natural history study and the work that Katherine's doing, those things will come together with the drugs. And I'm hoping to see, yes, we can identify this subset of patients need to be attached, and that's the language I really hope that we'll be using in 5 years when we're looking at which of the Stoke drug, which of the Dravet patients we should be giving it to. We're not going to give it to the Dravet patients that have that syndrome that Ingrid and Lynette Sadler discovered 2 or 3 years ago, because they've got gain of function mutations. You know, in those cases, that would be contraindicated. So I think it'll be a very different looking world at that time.
So Ingrid, what about you in 5 years? Where are you anticipating? You were there at the beginning, you're here now. In 5 years from now, what are you anticipating seeing? And just so that I can get them both at once, natural history study, we've just heard from Steve how important it is. From your view, how important it is, and how do we get connected? So 2 questions for you.
Well, I think it's critical.
Horizon. Yeah.
I think the natural history study is critical. I wouldn't be spending so much time and effort on developing it. But, and I thought that was beautifully shown by Joseph, where he showed the changes with life and just mentioned that we showed that 1/4 of women over about their teens get psychosis. So that was a whole new problem no one ever guessed about. And boy, is a psychiatric problem in an adult a huge issue. So I think that that's a nice example. And just to go back to something else, and I think something in the chat about gene therapy, when should you do it? Well, ideally you would do it in utero, but you won't know if most of these children have any disorder in utero. But that would be the sooner the better, because many of these proteins may be affecting brain development or networks in the brain. And so perhaps you could change something if you knew sooner. So that might seem a bit pie in the sky, probably is a little bit, but I think the earlier the diagnosis, the better. And Katherine's work on the acute diagnosis is really important. And Jenny said it worked for her, but we really need to get it around Australia, because at the moment it is at the big centres, but not at some of the other centres. And I was at a meeting this week, the Mercy couldn't get a baby diagnosed for about a month. So we really have to make that much more equal. So our services around the country. In terms of 5 years, better go back to the question.
Yes.
I think we might understand different types of gene therapy. We have to understand the functional effects, which is Steve's world. You know, what does it do? Which of those gene therapies get you back to the normal function of that gene, not too little and not too big? And I think that subtlety is huge. Also, it needs to be in the right cells in the brain. Not all cells are the same, they all have different functions. So we have to get the right cells at the right stage in development. You miss the boat, I think, often if you're looking at an older person. Having said that, I'm pushing the companies to consider trials in older people, because we don't know that they won't. That if you've got a Dravet who's 20 years old, how do we know it's not going to make a difference? It might. I don't know that it's realistic to think you'll end up with a normal everything, but you might improve their lives. So back to 5 years, I think we need to think about the additional genetic components. So although we're talking about these as single gene diseases, and Steve sort of said this as well, I think we have 19,000 genes. So the other 18,999 are influencing outcome. And we're part of a big European collaboration looking at about 700 Dravet patients, and they've all got SCN1A mutations. And now we're trying to say, well, what other genes are influencing it? Maybe this gene means you get the crouch gait, and this gene means your seizures go away, and this gene means your seizures are terrible, and your intellect is severe, or your intellect's mildly impaired. So there's a huge amount of work to do.
All right, Chris, I'm going to come to you if I may now. You're now there at the Epilepsy Foundation. Here's a question that's come in from our chat room. Is there going to be on-the-ground support, such as support workers visiting newly diagnosed families? So we've talked about, what Katherine has seen there at the Royal Children's Hospital, the pros and cons of giving a diagnosis to somebody when they're so young. Is there going to be any sort of support for families?
Well, the simple answer is yes. How we get there is a little bit more complex, but just hearing Steve, Katherine, and Ingrid talk, the diagnostic journey is much shorter. But in conversations with people in the 2A world, but also within genetic epilepsy community, that once our families are diagnosed, the support and the service around them haven't really changed since my son was born 19 years ago. So that is definitely a significant gap in services, and it's part of the reason we're doing the DEE survey. So we need this information to be able to advocate, and that will be part of my role at the Epilepsy Foundation. And the Epilepsy Foundation's broader role is to try and fill those gaps. How and what it looks like will evolve over time. It's not going to happen tomorrow, but certainly we want to make sure if someone rings into the Epilepsy Foundation through the national service that they are building, that we hope that once, if someone rings with KCNQ2 or SYNGAP, they are signposted again for those supports. And that's part of Emma's work. They're actually looking at what is the information people need once they're diagnosed. So we're trying to bring all those pieces together, but what it looks like and how we can make sure, to Ingrid's point, that someone in Alice Springs can get the same support as someone in Melbourne. So it's the answer is yes. What it looks like and how long it takes us to get there is still yet to be answered.
Well, Chris, I know you've only been there for about a month, so we're not going to put you on the spot yet in terms of the answer to that question. But I did want to ask numbers. I mean, we've reached through Twitter, 51,000 people today. I mean, that's just a tiny example of the kind of reach of this. And, we've had people on this call, not just from Perth, Adelaide, Brisbane, from the Outback, from here in Melbourne, from Sydney, but also from India, Nepal, the United States, Europe, the UK, and elsewhere. When and how are we going to convince the people in charge that this is the next cancer fight, that this is the next huge issue, medical issue that is affecting lives comprehensively for those who are born with these genetic epilepsies and for all of us who love them and who find that our lives must change because of those disabilities. How are we going to do that?
Well, if you're asking me, I think it's going to be a multi-pronged approach, but part of it is, I think, in the epilepsy world, we've been a bit shy about coming forward about our needs, and I think that's historical around what people viewed epilepsy to be. So it's not like you say, it's not something that gets a lot of empathy within the community, and I think we need to change that. And I think also, as we all have lived experience, the parents on this conference, our days are pretty full with just managing our children. So I think the investment that the Epilepsy Foundation is now putting towards this, it gives them an avenue for that voice. And we just need to make that voice louder. And to setting up GETA, we did that so that we could have a louder voice, because SCN2A on its own is not loud enough. So we need to bring all of these genes together and be a one cohesive voice to the funding bodies, whether it's government, whether it's through grant applications. We need to be together as one and advocating in a very collaborative way to make sure that those messages are getting through loud and clear, what our families are going through and the supports that they need, because it's just not good enough where we stand.
No, it's not. It's not good enough. You're absolutely right. And Danielle, that takes me to you, because you did that call out to government. You managed to get that $2 million. That was a fantastic start. You know, we have all benefited from what you did. That was not just for SYNGAP, that was for all the genetic epilepsies. When you look forward 5 years, what do you hope to see?
I'd like to be optimistic. I'd like to see clinical trials having been done for SYNGAP, and there being a viable treatment for SYNGAP, but for multiple genetic epilepsies as well. So I'm hoping that we're one of 10 genetic epilepsies that goes through this first round of finding treatments and finding the right people to use those treatments and access them. So that's what I'm hoping. And I couldn't agree more with Chris. This is the reason why we haven't gotten a lot of traction to date, is because we've been in silos, but GETA is part of the solution to that. Collectively, we're much stronger than we are individually. And
please go ahead.
I just wanted to give a big thanks to all the researchers, the clinicians, the scientists, even the basic scientists, which Steve calls himself. And also for Sarah James Day for hosting, but the researchers, we just wanted to say thank you, because you are our heroes. Honestly, we no pressure, but we're counting on you delivering for us. You really are our heroes. So keep going. If you ever need some inspiration, just need to ask us to bring our kids in so that you can see them and meet them. We are very willing to do anything you need so that you can progress towards treatments for our loved ones.
No, I couldn't echo that enough, Danielle. We have a couple more questions for our scientists. Any of you feel free to jump in on this. What should undiagnosed children do when it is believed that, or their families, I suppose, when it's believed that the condition is genetic, but nothing yet has been identified? Would you encourage them to get testing? I assume you would, but let me let you all answer that, Ingrid or Katherine.
I'm happy to jump in. Emma has been answering it magnificently on the chat, I can see. And Emma is a geneticist, so she is part of the group that really drive the genetic testing, although Katherine and I are hot on their tails. So basically anybody with a severe epilepsy, uncontrolled epilepsy, I think should have genetic testing. If you're on this call and you have not had it, there's a fantastic program called Beyond the Seizure, which is for under 60 months of age, where you can get what's called an in vitro panel done for free. Nothing's free in this world, and I'll explain why in a moment. But that just involves your doctor referring the baby from anywhere under 60 months of age to have this panel of testing. Now, this isn't the optimal testing, but it's a jolly good start, and it's got a lot of the epilepsy genes on it. The reason it's free, you know, there's nothing like a free lunch, there's no such thing as a free lunch, that's the same, is because the pharmaceutical companies are paying it because they want early diagnosis. And a good example of that is CLN2 disease, which is a very severe progressive degenerative disease. And a company called BioMarin has developed a very expensive precision therapy for it, which works, but they want to diagnose patients early so that they can get it early when it will work. So that's the reason this is available to us, but it doesn't mean we shouldn't be using it for every single child that has a seizure under 16 months of age. But if, and there've been a lot of other comments in the chat about what happens if you've had testing and it's negative, and that depends on the testing. So at the moment, the sort of clinical testing is called whole exome testing, which tests 1% to 2% of your genome that encodes your proteins. Now, at the moment, we are in the developmental and epileptic encephalopathies, we're solving 50%. So 50% of the children are getting a diagnosis, and adults, which is super fantastic, considering none were diagnosed 15 years ago. But what about the other 50%? And one of the recurrent questions is, can I get my exome tested again? So it's not the panel I was talking about. This is whole exome sequencing. And the answer is yes, it can be tested again. And Emma is closer to this, but it looks like she said every 18 months or so they can talk to it. I don't know if I've got that quite. They can review it to look for new genes, because new genes are being found every week. I'll let Katherine add.
All right. Okay, Katherine, I'm going to ask you a slightly different question because we have 6 minutes left, and I want to give each one of you 1 minute. I'm going to ask you to really try to hold to that, and then that gives me about 60 seconds to say goodbye. So, Katherine, I'm going to start with you. If you have final thoughts, would you just let us what your thoughts are as we're, you know, summing up and leaving today?
I think it's been a wonderful meeting. Thank you to the GETA team for organising. It's been fabulous. I think that we as clinicians learn a lot from this kind of thing, you know, too. And it was wonderful with the breadth of knowledge and experience shared today. I think this is really important for helping us move forward, that everybody gets together to do that, and to thank you all for driving that over the last 5 years.
Steve, you have 1 minute. What have we missed? What's important for families to know about the research you're doing? What comes next?
Well, two quick things. One is, I think we've come a remarkable long way in 5 years. When you see almost every speaker today had therapy in their language. And that's a really, really big change. So I think that the one big thing we want to give to the families that even though from year to year it looks small, that there's great hope, and that I think you'll look back and we look back at the last decade and you'll see what remarkable progress we've made. So I think everyone should come away from this feeling positive and not feeling alone.
All right, Ingrid, I'm giving you your last minute.
I think we've, firstly, congratulations, GETA. Fantastic job, as always. I think you illustrate the importance of us all working together as a team, and I feel that's happening much more nowadays, and that patients and families are much more of an, you know, regarded as a major voice. I think precision diagnosis is the first step, and that's where all the gene testing comes in. But then, you know, I always say that the gene is the beginning of the answer, and then we're headed towards precision therapies. We need to make sure these gene therapies work. And the Tim Yu story has had a lot of coverage everywhere, including the New York Times, but it's not a rigorous trial, and we need to have rigorous trials. So thank you all for working with us in that space, so that in 5 years' time, we know that what we're giving people works.
Danielle, let me come to you if I may. Do you have any closing thoughts for us?
Sorry, on mute, standard quote for 2021.
That's all right. That's all right. You've only used 10 of your seconds.
I've got 2 minutes running around outside, so I'm trying to
That's
temper
the
it.
life of it, isn't it? Please do.
So yeah, hope. Biggest for me, I look forward to the GETA conferences because it gives me hope, and also in this COVID world, we haven't had that exposure that we always usually do of the other conferences. So it's really nice to see that there's a lot of success that's happening in precision medicine. Therapies are being accomplished, and people are getting better from them. So hope and connection. That's the only thing I just wanted to make sure that we stay connected, and that we express our gratitude to all the clinicians, researchers, scientists, and everyone who brings this all together for us. Thank you.
Well said. And Chris, if I can give you a minute as well, there's a lot that you've been doing in your various roles with SYNGAP, but also with the Epilepsy Foundation, your new role. What are your thoughts as we close out today?
One of my observations really from all the speakers is that everyone talks about the family's voice, and I think that we've actually progressed a long way with that since I've been involved in. So I would encourage families that are on this call to have a voice and make sure that they share their stories, to Danielle's point, and really work in a collaborative way. So if you're doing something in your gene, reach out and share that with someone else. It's the way we're going to move forward much faster. It really makes a big difference if we can share our learnings. And yeah, I agree. I think we've got hope and change. I guess that's my other word. I'm looking for change. So let's keep progressing that forward.
Yeah, I'm looking for change too. And I feel like this is a team that's already accomplished so much, and I don't have any doubt that more will be accomplished. And I don't have any doubt because of the extraordinary scientists, researchers, clinicians, doctors, therapists who are on this call and who have given us their time here in Australia and elsewhere around the world. I don't have any doubt because of all of you who've joined this call. You have busy lives. You know, Danielle was just talking about her kids running around. I love my daughter Jackie dearly, but I outsourced her. And thank you, Andrew, for having Jackie up at the farm collecting hay with the 5K lockdown. It's slightly farther than that. Shh, don't tell anybody. Chris, I know you get this too. The only way we're going to do this is together. We are thrilled about the fact that all of you have individual foundations, and if you don't, you have GETA. But we also think, regardless of even if you have a vibrant foundation, it is important to connect. They're called genetic epilepsies. We've heard about all the comorbidities, the other things that come along with these epilepsies. Together, we can seek more treatments. We can get precision medicine steamrolling ahead. Actually, I'm going to drop the steamrolling analogy as something from yesteryear. I want to be like Elon Musk and basically send it to the sky in a rocket. And I'm going to count on all of you to be the boosters for that. So thank you very much for taking this time to be with us. We know you're busy. We appreciate that you joined us. For us here at Genetic Epilepsy Team Australia, I'm signing off now. Thanks again, and have a good day or a good evening wherever you are in the world.